Hello students, welcome to the next week of the SWAYAM course Anatomy of Flowering Plants. In this week we will be discussing about the tissue system. So previously we have talked about various types of tissues and how they are arranged to form a simple layers or to form a complex layers. In this we will try to find out how different types of these tissues are also arranged to form a tissue system. We will talk about epidermal tissue system, then ground tissue system and then vascular tissue system. So first of all, let us start our discussion with epidermal tissue system. So epidermis is definitely the outer layer of the cell or the outer boundary of all the organs of the plant. Basically we call it as epidermis when we talk about above ground organs. When we talk about roots, then it is actually also an outer boundary, but the name is slightly different. We call them rhizodermis or epiblema or piliferous layer. So that is also there in the underground organs. Now coming to epidermis. Now epidermis is derived from protoderm by anticlinal division and they are living cells. See, when we were discussing about apical meristem, suppose we are discussing here, we are drawing here and then about the shoot apical meristem. Okay, and then we have also studied about root apical meristem. So this is the region of root apical meristem. So in this also we have different layers. Okay, so the outer layer will give rise to the epidermis, which is the protoderm. So here also you have different layers. So the outer layer will give rise to it will be formed from the protoderm. These are living cells. Basically you can call them as parenchymatous cells and they are living, thin walled, made up of cellulose, and this protoderm is formed, this epidermal layer is formed by anticlinal division of the protoderm. So this also when we were discussing that the outer layer the surface layer, so it is dividing anticlinally. So in this way if it is dividing, it is increasing the number of cells on the either side. So this type of division is anticlinal, perpendicular to the surface, clinal. Okay, that means perpendicular to the surface. So it is increasing the number of cells on the surface, it is increasing the surface area. So this is basically the epidermis. Now it is found in all the parts of the plant like leaves and flowers and stem, but in stem when the secondary growth will happen, this epidermis will be replaced by periderm. So that also we will discuss later. In this, cells are compactly arranged with no intercellular spaces. Suppose this is the outermost layer, okay, and this is the epidermal layer. Now you can see here that the cells are compactly arranged and they don't have any intercellular spaces, because if spaces are there, so they won't act as a proper protecting layer or as an outermost layer. Generally the epidermis is uniseriate, means a single layer of epidermis is generally found in most of the organs. However, in some cases it can be multiseriate, like 2 or 3 epidermal layers can be there, but it's not very common, like in case of some roots of orchids, okay, which are epiphytic plants, then in ficus also, in nerium also, they are multiseriate. Now how they become multiseriate? Definitely by periclinal division. Suppose this is the outermost layer. In this way, okay, parallel to the surface, if it is parallel, then they will give rise to cells on these sides, and by which they can become multilayered. Now most leaves, if we talk about leaves, they have 2 epidermis. Like if we cut the section of a leaf, if we cut the transverse section of a leaf, we can see here that 2 epidermis is present. Otherwise, the stem have a circular boundary. So, in that case, you can say that it is single layer, and in some other leaves also, suppose some circular things are there in circular orientation, so they will have a single boundary, but in leaf, upper epidermis and lower epidermis. Like tubular leaves or those which have a circular boundary. So, like tubular leaves, onion, leeks and garlics, in monocot plants generally we can say that they have 1 epidermis. In stems also you will find an epidermis which is similar to that which is found in leaves. That means it will also have various other components which are characteristic feature of leaf epidermis. Now, coming to the function of these epidermis, see basically they act as a boundary. So, you can say that they are also protective in nature. They are protecting the inner parts of the plant from outer environmental cells. They are protecting them from any other biotic or abiotic stress like some fungal attack or bacterial attack. So, they cannot, these pathogens cannot pass this epidermis layer. Okay, so they are acting as a boundary. Similarly, like some high UV radiation is there or high light intensity is there. So, in that case also some protection could be provided by the epidermal layer. Then from mechanical injury, if some injury is there, so it has to cross the epidermis boundary. It also protects the plant from loss of water through transpiration. Means you all know that plants are transpiring, they are taking the water from the soil and that is lost from the surface, from the leaf. Basically, we will study that in leaf stomata are there from where the plant water is lost, but excess loss of water is definitely harmful for the plant. So, the epidermis allows that the water is not lost too much if that boundary is not there, because it is a boundary of completely compactly arranged cells with no intercellular spaces. So, the loss is not very rapid. It can also act as a storage organ. Sometimes it also helps in secretion of many of the compounds to the outside environment. It also performs photosynthesis. See, actually photosynthesis is basically not the role of epidermis, but yes, in epidermis you have stomata, and stomata have guard cells, and which we will see that guard cells have chloroplasts. So, they can also perform photosynthesis. Sometimes some pigments may also be present on the epidermal cells in the vacuoles of the epidermis. So, like anthocyanin is present. Anthocyanin is a pigment which is found in the vacuoles or the cell sap of the vacuoles. So, they will give color. Like some flowers you would have, in some case of flowers or fruits, sometimes the color might be there because of the presence of the outer layer. Now, coming to the components of epidermal tissue. See, it is made up of pavement cells which are the basic epidermal tissues. Okay. Now, stomata, cuticle and trichomes. Now, when we talk about epidermis of roots, it is actually we do not call it as epidermis, we call it as epiblema or piliferous layer, or even we call it as rhizodermis. Okay. They are, you can say that they are specialized form of epidermis, and we can differentiate between them in this way that they give rise to unicellular hair, this rhizodermis. Okay. They give rise to unicellular root hairs. Now, this root hair is basically extension of this epidermis. Okay. Cell, extension of the epidermal cell. So, always root hairs which are unicellular in nature, and you can say that they are extension of the epidermal cells, they come out from the roots. Root epidermis, and they increase the surface area for better water and mineral absorption. Another important difference is that root epidermis do not contain stomata as well as cuticles. So, that all we will see that what are these structures and how they are affecting the function of the epidermis, but they are not found in roots. Now coming to the pavement cells. Now these pavement cells are the basic epidermal cells or the most abundant epidermal cells. You can say, suppose this is the surface of a leaf. Suppose you imagine this is the surface of a leaf, and you will find lots of epidermal cells. Okay. So these are also known as pavement cells. Like apart from stomata and trichomes and cuticle, which is a chemical compound, all the rest of the cells are known as pavement cells. Now when we observe it from the surface, we can see that they are tightly packed with no intercellular spaces, and the pattern also vary, like different pattern in which they are oriented, and different shapes which they are having, different sizes which they are having, that is also variable. And if it is observed, generally they do not have a regular shape, and they are quite irregular in their shape, and it is found that they are arranged in a zigzag puzzle type, which helps them to interlock. Like if you find a piece and you can fit it between these cells, so they can interlock in that way, and this orientation will help them to reduce intercellular spaces to have a more compact arrangement, so that they can act as a protective tissue. So definitely they decrease water loss. So that all we have discussed before, the role of epidermis. So we are actually talking about the pavement cell in many of the cases. See, they maintain internal temperature. See, because they are so packed and they don't have any spaces, so they help in maintaining the internal temperature, because the external environment may also not have effect to the internal tissue because of these pavement cell. They also help to keep the inner cells in place. They also provide protection. So, okay, they also separate stomata from each other. Now, two stomata could be separated by these pavement cells so that they are not clumped together. So, you can find pavement cells between these two stomata. You can see here a lot of different variation of pavement cells are shown here. Different structures are there, and stomata also you can see dispersed in this pavement cells. Now coming to the most important part, which is or structure which is present in this epidermal cell, is the stomata. Now stomata is basically a pore which is found in the epidermis. When we draw the structure of this stomata, we will see here So, when we will observe the structure, we will find that stomata is having a pore, okay, stoma, that is the pore, and it is surrounded by two guard cells. Now, stomata is found on the surface of leaves and young stem. Okay, these guard cells actually help in controlling the opening and closing of stomata. So this is the structure which is showing an open stomata, and you will have closed stomata also. See, when the stomata or the opening is closed, it will look like this. This is swollen, okay, or you can say turgid, and this type of stomata is known as flaccid stomata or shrunken. Okay, so here also you will have nucleus and lot of other organelles. So this guard cell, when they absorb water, they swell, and because of this swelling they become more turgid and they open a pore in between which is known as stoma, and when they are closed, when they lose water and water is not present, they are not so turgid. So in that situation we call them as flaccid and they are shrunken and the stomata gets closed. Now stomata is very important for gas exchange because when plant or when the leaves are performing photosynthesis, they require CO2, they require means oxygen exchange is there, carbon dioxide exchange is there. So all those gases exchange is there and it is all required for photosynthesis. So it's required that stomata should open, okay, and in photosynthesis definitely takes place in the presence of light. So when light is falling or when during daytime, it's required that stomata should be open to allow these gases exchange. Now this stomata, they also help in water loss through transpiration, okay. When this also know that this is also a point where water will also move out through stomata. So the plant will decide that under what situation it has to open the stomata and where it has to close it. So all those factors will also be decided and that type of arrangement or that type of modification will be made by plants. But for in normal condition, during daytime the stomata is open to allow gases exchange. Mostly this stomata is found on lower surface of leaf just to reduce transpiration, because on lower surface of leaf there is more shade, there is more humidity maintained. So water loss in the form of transpiration will be low. So if stomata is found on the lower surface of the leaf, okay, so definitely less water will be lost, because in more drier condition more water loss is definitely occurring. It is closed at night and it remains open during daytime. Now how actually this stomata is opening and closing, in short we will try to understand. See, it is regulated by the movement of guard cells. Now these guard cells, what happens? Now as we can see here, the thickness of the guard cell is variable. We can see that they are more thickened on the inner side, on the pore side, and this thickening is more because of the presence of more cuticle deposition, and this cuticle is more permeable to water. Now what happens? They also have radial wall thickenings. Cellulose are there, microfibrils of cellulose are there, which are oriented in radial direction, radial pattern. So all this leads to different thickening pattern in the guard cell. Now this guard cell have a photosensitive receptor which is known as phototropin. Now phototropin is sensitive to blue light. So what happens during the daytime when light is falling, this blue light activates this receptor, this phototropin receptor, and this receptor once activated, now with the help of ATP which is produced during photosynthesis, what it does, it pumps proton outside. Now once these protons, positive ions are pumped outside, the a negative potential is generated on the inner membrane side. So what happens, because of this negative potential, lot of potassium ions and lot of nitrate potassium ions come and accumulate here. H plus ion also comes and accumulate here. This sucrose which is present in the guard cell, they also split up, and they also, starch is there which splits up to form sucrose, okay. So lot of ions accumulate inside the guard cell, lot of metabolites, a lot of solutes, they all accumulate in the guard cell, and because of this the concentration of water decreases in the inner side of the guard cell, and because of which water from the outside, from the other epidermal cells, they enter inside the guard cell, making the guard cell turgid. So when any cell will absorb water, they will become turgid, they will swell, they will try to stretch their walls, push their walls. So they will push their walls. So both the sides of the wall will be pushed, but the inner side of the wall, because it has thick cuticle deposition, it has thick cell wall, it won't stretch on this side, it will try to bend and stretch on the same side it's where the outer wall is stretching. So in that way it will leave a pore in the center. So and when water will definitely come out, this whole thing will be reversed and finally the cell will become static, flaccid. Now stomatal, this stomata, the number of stomata which is present on leaves, they will also vary, which is actually then counted also. It is per unit area basis it is counted, and it varies from 100 to 1000 per centimeter square. Now this we have discussed about that how the guard cell is helping opening and closing of stomata. This is also regulated by various other conditions also, like humidity, presence of some growth regulators. So these all factors combine together to open and close the guard cells. Now coming to the subsidiary cells, means these guard cells are surrounded by other epidermal cells, okay, other epidermal cells. So these other epidermal cells are known as subsidiary cells, and they will, and together with the stomata they form a stomatal complex. Now how this stomata is arranged and these other subsidiary cells are arranged, this has different nomenclature, and the study of these stomatal complex is known as tomography. Coming to anomocytic type, when we say that the organization of this stomatal complex is anomocytic type, in this the stomata is surrounded by variable number of subsidiary cells, and these subsidiary cells are almost all alike. Okay, in this all these subsidiary cells are alike and their number can vary. Now when we talk about anisotype, cytic type, in this stomata is surrounded by 3 accessory cells, of which one is distinctly similar than the other 2, means 3 cells, okay, surrounded by 3 cells, and we if we observe them, we will see that one cell is smaller and the rest of the 2 cells are bigger in structure. So this is about anisocytic type. Parasitic type is parallel celled. In this type of stomata, it is surrounded by 2 subsidiary cells or accessory cells which are parallel to the long axis of the pore. Suppose this is the stomata. So it will be surrounded by 2 sets of these subsidiary cells, and which will be parallel to the pore axis. Okay, so this is parasitic type. Coming to dicytic type, in this stomata is surrounded by a pair of subsidiary cells whose common wall is at right angles to the guard cell. They are the common wall, this is the common wall and this is at right angles to the guard cell. So this is the subsidiary cell. Graminaceous type, in this type of, it is found in monocot, in which the stomata is dumbbell shaped. So in dicot it was bean shaped or kidney shaped, and in this it is dumbbell shaped. Okay. So this is the guard cell, and these are the subsidiary cells. Okay. So it is dumbbell shape in monocot, and when we talk about dicot, we were that was bean shaped or kidney shaped. Coming to floating type, in which a guard cell is surrounded by a single subsidiary cell. A single subsidiary cell is surrounding. Tricytic, in which guard cells are surrounded by 3 subsidiary cells. Tetracytic, in which they are surrounded by 4 subsidiary cells. Now, it is also classified on the basis of ontogeny, means their origin, from where they have coming out. So, mesogynous type, if the guard cell and the neighboring subsidiary cells have common origin. Then the developmental pattern is known as mesogenous. It is mesoperigynous, in which some of the cells have common origin. Some cells have common origin, whereas not all. So, in that case it is known as mesoperigynous. Perigenous type, when there is no common development between the neighboring and subsidiary cells with the guard cells. So, no common origin between the subsidiary and guard cells. So, that condition is known as perigenous. Stomata vary in distribution and depth. So, basically when we talk about like the surface of the leaf, upper surface and lower surface, okay. So, it has basically leaf has 2 surfaces, the upper surface and lower surface. Upper surface is known as adaxial and lower is known as abaxial. So, the distribution of stomata is also not uniform on both the surface. We call it as epistomatic if stomata is found only on the upper surface, like in floating leaves. Okay, so stomata, definitely in summer species stomata is not found. So, it is always found on the if the leaf is floating, it will be found on the upper surface. We call it as hypostomatic when leaves with stomata only on the lower surface. Like in Nerium, it has modification. This type of pattern is found in Nerium plant because this pattern actually helps to reduce transpiration, to reduce water loss through transpiration. Amphistomatic, in which leaves have stomata on both the surface. Now, monocot leaves are often oriented in a vertical fashion. So, we do not call them having any lower or upper surface. So, typically we say that they are amphistomatic, that means stomata is present on both the surface. Now, in dicots, generally they have stomata on both the surfaces, so we can call them as amphistomatic, but generally the stomata is found more on the lower surface. This is also to reduce loss of water through transpiration, because lower surface will have a more shady environment, will have more humidity, will be less affected by the light and temperature. In monocot, the stomata will be arranged in linear rows. Suppose this is a monocot leaf. In this, you will have stomata in linear arrangement. Suppose this is a monocot leaf. You can make it stomata like this. Linear arrangement. And when we talk about dicot leaf, then it has irregular distribution of stomata. Or you can say scattered. Stomata is scattered in dicot, whereas in monocot they are arranged in linear fashion. Now this stomata, wherever stomata is present, it opens on a substomatal cavity which is filled with, which is actually a space inside the leaf where actually gas exchange takes place. In xerophyte, you will find sunken stomata, so that we will discuss when we talk about xerophyte, and in hydrophyte, stomata will be completely absent. So this is all about today what we have talked. So next we will cover the other components of epidermis, like cuticle is there, presence of trichomes, so that we will take on in the next class. Thank you.